Hi, my name is Charlie Dvergsten, and I'm a biology major and biochemistry minor at Carleton College. I'm Dr. Robertson's REU student in a marine toxicology lab focusing on secondary metabolites of benthic dinoflagellates. These little algae are very important to both humans and fish, as they give humans oxygen, fix carbon, and are primary producers. But they also produce a host of secondary metabolites, some of which are very toxic. The illness caused by these toxins is called ciguatera fish poisoning and is very prevalent in tropical parts of the world. My project focuses on secondary metabolites produced by these algae, more specifically how they contribute to toxicity in fish and mammals. To do this, I'm growing algae cultures and focusing on two bioassays, the erythrocyte lysis assay and the acetylcholinesterase inhibition assay. Here are my algae cultures. I am growing 5 different species across 4 genera. These are important because there are differences in toxin production at different stages of life for these dinoflagellates. These toxins, mainly ciguatoxin and mitotoxin, are very large molecules for unicellular organisms to produce. Mitotoxin is actually the largest compound made by any unicellular organism. Because the toxins are very biologically expensive to produce, they are made in different amounts through different growth phases, primarily exponential and stationary growth phases. The first assay, the erythrocyte lysis assay, measures hemolytic activity caused by algal metabolites. This assay will mainly indicate the presence of mitotoxin, or another unknown metabolite that causes hemolysis. Mitotoxin affects calcium channels, starting a cascade that ends in lysis. Here is an example of a 96-well plate for my erythrocyte lysis assay. Darker wells show blood cells that have lyzed, whereas lighter wells have more intact red blood cells. The acetylcholinesterase inhibition assay will test neurotransmitter balance and signaling when these toxic metabolites are present. Ciguatera fish poisoning often involves neurological symptoms, so this assay will indicate the presence of toxic metabolites that potentially contribute to neurotransmitter inhibition. A normal day for me right now involves setting up some 96-well plates to test for red blood cell hemolysis, counting my algae cultures to obtain growth curves and growth rates, and doing some dishes. But that changes every day. When my algae cultures finish, I'll be extracting toxins from them, running the acetylcholinesterase inhibition assay, and starting to write my paper for this project. Another great thing about this job is that there are a lot of atypical days. For instance, yesterday I just spent 10 hours on a boat, got to go snorkeling with Dr. Robertson, and help set up a completely new project. Thank you for watching. I hope you enjoyed learning about my summer research.